Welcome to Med Together. This is a podcast by the people, for the people, as it were. My name is Chana. I'm a med student in New York. This podcast deals with a lot of med school topics, starting from the basic sciences and building its way up, and hopefully it'll be a bit of review that can help y'all consolidate and synthesize important information and maybe phrase it in a way that's easy to understand. By the way, if you're pre-med or you're a nursing student, or you're just interested in sciencey, medically topics, and you're not in healthcare at all, you're totally welcome to. We don't discriminate. If you're interested, you're cool. Ready to rock? Let's do this. Hey everyone, welcome if you're new. Welcome back to my intrepid repeat listeners. Today's talk is in the cell biology series, and we're going to talk fairly briefly about some basics about how we go from an amino acid chain, which we discussed in the DNA RNA lecture, to an actual full-on protein that works, and also some QC measures that we have in place for when things go wrong. So what is a protein? Proteins are translated from RNA, which is transcribed from DNA, which we know. But when it comes off the ribosomes, it's a linear chain of amino acids, which we call the primary structure, and that is not a protein. It needs to be folded in order to be a functional protein. Structure and function are completely interconnected, and you cannot have function without the proper structure. So the central dogma of DNA to RNA to proteins is really incomplete if we don't talk about folding. So what actually determines the folding structure of a protein, and how is it that the same proteins are always or almost always folded in the same way? A lot of it is determined by the amino acid sequence itself, the hydrogen bonding, the hydrophilic and hydrophobic residues, disulfide bonds, etc. And the way we figured this out was by a series of really interesting experiments by a guy called Anfinsen. And Anfinsen determined that if he took a protein and he denatured it and he left the denaturant in place, it would refold randomly because the denaturant, he used urea, disrupts hydrogen bonding and it makes the hydrophobicity a lot less important, essentially. But he found that if you denature a protein and then you put it in a solution without the denaturant, it will mostly reform correctly, which indicates that there's some folding pattern dictated by the amino acid sequence, and it's capable of finding its own most thermodynamically stable form in a natural environment. So there are proteins that we can think of as two-state proteins, which are either folded or unfolded, and there's kind of nothing in between. The unfolded state is higher energy, the folded state is going to be its lowest energy, so it's going to move towards that thermodynamically. Other proteins, and this is really most proteins, are going to fold in a more complex way, where they have intermediate states that might be slightly lower energy than the unfolded state. And this is important in protein misfolding, because sometimes the proteins can get stuck in these little low energy pockets, and then they don't continue to fold. The major driver of folding is hydrophobicity. The hydrophobic residues need to be protected from the outside. And if the hydrophobic parts are exposed during folding, like in an intermediate state, it can cause protein aggregation of multiple proteins because they're trying to bury the hydrophobic molecules. This brings us to a very important class of proteins that are important in protecting other proteins from misfolding, and these are called chaperone proteins. Chaperone proteins help proteins fold properly, and they protect them from aggregating with each other or with other cellular elements and from misfolding under various stresses. And they start interacting with the protein as it's coming off the ribosome before it's even fully translated. And they are extremely abundant, and we'll see why in a minute. And chaperones selectively target unfolded proteins by recognizing exposed hydrophobic residues and binding them in a little hydrophobic pocket that protects them and allows them to fold properly. I want to talk specifically about HSP70 chaperones. HSP just stands for heat shock protein. This is a critical class of chaperones. They were discovered first in bacteria, and they engage the nascent chain and work with co-chaperones HSP40 to help it fold properly. These proteins are made up of an ATP hydrolyzing domain, and then a substrate binding domain that has a beta sheet subdomain and an alpha helix subdomain, which kind of acts like a lid. So the space between the sheet and the helix is like a little pocket, and it can hold about 6 to 8 amino acids. So you're going to need multiple chaperones per protein, as you can see, because proteins are many amino acids long, and this chaperone pocket can only hold about 6 to 8 amino acids at a time. So the ATPase domain controls the opening and closing of the alpha helix lid. So it hydrolyzes ATP to ADP slowly, which closes the active site. And then there's an exchange factor which opens the ATP hydrolyzing site by exchanging ADP for ATP again, and then the substrate is released. So it's open when it's bound to ATP and it's closed when it's bound to ADP. And then there are co-chaperones that work with the heat shock proteins to direct them to the correct substrates. And there's a lot we still don't know about this family. This family and their co-chaperones are a very complex network of folding machines, which are utilized by cells in many ways. And despite considerable progress in the elucidation of the mechanistic basis of these machines, there are important things that we still have not solved. With respect to the HSP70 proteins, it's still very unclear whether their activity to assist protein folding relies on the ability to induce conformational change in the bound substrates, or if they're just protecting them, how the coupling mechanism allows ATP to control the binding, and to what extent the sequence variations within the family translate into variations in the mechanism. So all that we really don't understand, we just know that they're important. And with respect to the co-chaperones, we really don't have a good molecular understanding of how co-chaperones target their HSP70 partner proteins as substrates. And we expect that there are more cellular processes that are dependent on the chaperone activity that we don't know about yet. So we still have a lot to learn. But these are very cool and very important proteins. But of course, sometimes things do go wrong, even if you have chaperones. So what is our mechanism for getting rid of proteins when it goes wrong? So essentially, we have to be able to degrade misfolded proteins because, and we'll talk about this a little bit later, if we don't degrade the misfolded proteins and they're allowed to aggregate, this causes a lot of toxicity to cells. So the way that cells deal with these misfolded proteins is by either autophagocytosis or proteasomal degradation, mostly. So autophagocytosis can happen through macroautophagy, where the cell basically builds a vesicle around the misfolded protein and then delivers the whole thing to a lysosome. It can happen via microautophagy, where the lysosome kind of like eats off pieces of it, like bit by bit. Or we can have chaperone-mediated autophagocytosis, where the chaperone can recognize that the protein that it's trying to help is too far misfolded, and the chaperone will actually deliver it to the lysosome. And then there's a protein on the lysosome which translocates it inside for degradation. There's another kind of mechanism of degradation besides for lysosomal degradation, which is proteasome degradation. So in this case, proteins that are misfolded undergo ubiquitination, and this can happen when they're damaged or misfolded, or it can also happen when the cell is just done with it. It doesn't need this protein anymore for whatever reason. So they get tagged by polyubiquitin, which is usually a tetraubiquitin, but not always, and they're going to be recognized by the proteasome. Now the proteasome is like a pore structure with active sites inside, and the entry is gated by ubiquitin. So only ubiquitinated proteins are allowed in, and once it starts to be allowed in, the chain kind of gets unspooled and it gets fed into this narrow channel by ATPases on the top of the proteasome. And inside the proteasome, there are these active sites which degrade it. Now, to understand the importance of these QC mechanisms, we have to talk a little bit about why protein misfolding is so important and so damaging. So misfolding can be disease-causing in a gain-of-function manner or in a loss-of-function manner. So loss-of-function manner is maybe easier to understand. It's fairly simple. If they are misfolded and they can't do their function, the cell is lacking a protein that it needs. So cystic fibrosis is a prime example of this. The cystic fibrosis transport channel is a chloride channel that doesn't work properly when it's misfolded. So this is one of these proteins that actually comes off the ribosomes into the ER and gets sent to the membrane. Next talk, when we talk a little bit about how proteins are trafficked, we'll discuss this even further. But this is a really, really complex protein. And it's so complex that actually even in normal people, most of it doesn't fold well, and a lot of it ends up getting wasted, probably close to 80%. But when there are mutations, it can disrupt the interactions with its chaperones, so it gets misfolded even more frequently, leading to even less functional protein. More like only 1% of this protein turns out functional in people who have CF mutations. And this understanding of why the mutation causes this problem is interesting in terms of drug development, because there are new ideas of having small molecule drugs that act as a chaperone to help it fold better, perhaps in people with cystic fibrosis. Now, another example of a disease that can be caused by loss of function is emphysema. Emphysema is a problem where you have basically destruction of the alveoli in the lungs. And what happens is there's a protein called alpha-1 antitrypsin, which is a protease inhibitor that gets synthesized in the liver, and then it inhibits neutrophil elastase in the lungs. And it works by trapping the protease in a covalent complex. So it's actually kind of a cool thing to see. It baits the elastase with a sequence similar to its target, and then when the protease binds and thinks it's about to degrade something, it has a domain that swings around and snaps shut, locking them together, and then the whole complex gets marked for the proteasome. So it's like a little suicide inhibitor. And mutations can affect alpha-1 antitrypsin in several different ways. So first of all, it can make it target the wrong protease, or it can make the trap swing shut too slowly so it can't catch the protease, or it can be hyperreactive and it can get tripped without the protease at all. And so in any of these kind of mutations, you can understand where you might have a problem. You can't get rid of this neutrophil elastase. You have too much elastase and it starts eating up the alveoli, and that's how you end up with emphysema. Now, alpha-1 antitrypsin mutations are interesting because there are a bunch of different mutations, and they can actually also exhibit gain-of-function protein mutation issues. What do I mean by that? So there's a specific mutation, which we call the Z mutation, which allows intermolecular interaction between alpha-1 antitrypsins in that trap domain, and you end up getting a chain of alpha-1 antitrypsins that all trapped each other. So they kind of build up in this long aggregate. And proteins with this mutation, besides being non-functional, actually can't even be secreted from the liver because of this long chain that it's formed, and it builds up and can cause cirrhosis in the liver. So this is a gain-of-function problem where not only do you not have the proper function in the lungs, you also have the fact that you have aggregates causing damage in the liver. In general, gain-of-function issues in protein folding are mostly mediated by protein aggregation. So some more examples are like Alzheimer's. So in Alzheimer's, you have an amyloid beta peptide, which is a fragment of an amyloid precursor protein. And these fragments aggregate and cluster near the neurons, causing damage to the brain. These amyloid beta peptides have a very stable structure of hydrogen-bonded beta stacks, and we call them amyloid fibrils. We have found that in some hereditary forms of Alzheimer's, there are mutations in the amyloid precursor protein, which lead to more aggregation causing. In Parkinson's, we have a similar situation. Parkinson's is caused by death of dopamine-producing neurons. And the reason why this causes the symptoms of Parkinson's is because dopamine controls a lot of the motor control in the basal ganglia. It's actually fairly complicated and extremely fascinating, but suffice it to say that if you don't have enough dopamine, the basal ganglia inhibits the thalamus and inhibits movement, essentially. So you get slowing, you get tremor, you get rigidity in the muscles. And this is why we treat it with a medication which we call levodopa, which tries to replace dopamine and works temporarily. But the mechanism of the disease that causes the death of the neurons is similar to Alzheimer's. There's a protein called alpha-synuclein, which gets misfolded and it starts to aggregate and it forms fibrils called Lewy bodies, and that causes the death of the neurons and the disease. If you have Lewy bodies in places outside the basal ganglia, it's called a Parkinson's plus syndrome, Lewy body dementia, and you can see cognitive changes as well. Now, if we're discussing misfolded proteins, we have to talk about prions. Prions are really interesting because they're amyloid fibrils that are transmissible and they cause other proteins to misfold. They're almost like contagious misfolding. We don't really know why or how this happens, but we know that there are different kinds of amyloid fibrils which are associated with different strains of prion disease. And more recently, scientists have been thinking that neurodegenerative diseases like Alzheimer's and Parkinson's share some characteristics with prions because both of these diseases are propagated in a prion-like fashion. You can see the transmission of the disease between cells, and the degeneration tends to follow neural connections. So there has been also a lot of research into thinking about trying to maybe block the transmission of Alzheimer's or Parkinson's between neurons. So if you could catch it early, maybe you could stop the spread, if not reverse it. So lots of interesting applications to a lot of these basic science ideas, which is why they're so important and so fundamental and so foundational. So as promised, short and sweet, talking about protein folding, which is really important for functional proteins. Folding is governed first and foremost by trying to hide away any hydrophobic domains. And a lot of protein is helped along by chaperone proteins. Improper folding can lead to loss of function like in cystic fibrosis or deleterious gain of function like Alzheimer's, Parkinson's, etc. Improperly folded proteins should be recognized and destroyed by lysosomes or proteasomes. Thank you so much as always for listening and for sticking with me to the end. I hope you enjoyed and you learned something new today, and I'll catch y'all next time.